this video we will discuss about how this STING agonist DiaBZl will impair the tumor growth, and this effect will be enhanced along with this T cell receptors, T cells or engineered T cell receptors, that is 1G4 TCR-T receptors are engineered with the T cells, and when they use these T cells along with this DiaBZl, it will enhance the tumor suppressions on. And they did the experiment in the mice model, in vivo experiments in the mice model, where first they inject the cancer cells, that is MEL 526 cells, and then they build the model for 1 to 2 days. After that, the tumor development occurs from day 3 to 12, and thereafter they treat the mouse with the DiaBZl and this engineered T cell receptors, and thereafter they observe the tumor volume and the tumor size, and they did it for different conditions. And what they have find out is that compared to the mock, where they have not injected any STING agonist or engineered T cell receptors, they find out that when they use the T cell receptors that are treated with the DiaBZl, and the DiaBZls also in the mouse models, they see that maximum reductions in the tumor volume occur compared to the other cases where only the T cell receptors that are treated with the DiaBZls are introduced. They see that there is a reductions in the tumor volume, but it is higher than the combined case. And when they use only the DiaBZl, the effect further goes down. And when they use only the T cell receptors that are not treated with the DiaBZls, it effects is further goes down, and it is more close towards the mock results. And same things they have observed in the panel C. They have shown the tumor size, where we can see that in the cases where engineered T cell receptors and DiaBZls are introduced, they see that minimum tumor growth, whereas in all the other cases the tumor growth are higher. And uh, uh, and the case where the T cell receptors are used with the DiaBZls, the tumor volumes are similar, almost similar to the control case or the mock case. And the same things is shown, and the same things are shown. Similar things are shown in the case where this T cell receptors that are treated with the DiaBZls shows that T cell receptors. This is the difference between panel B and panel C. In the panel C, we can see that this conditions, that is T cell receptors with the DiaBZls, tumors rate. So this first conditions is that engineered T cell receptors for the DiaBZl. Second one is only the T cell receptors. So this only T cell receptors shows the result close to the mock, and same things are observed in the panel C, where the T cell receptors shows the result close to the mock. And when they check the tumor weight, they also find the tumor weight results are also in line with the tumor shape. That is, when they use the DiaBZls and DiaBZl treated engineered T cell receptors, they see that minimum tumor growth. However, for the case where only the T cell receptors without treated with the DiaBZl used, they see that the tumor growths are reduced to a lesser extent compared to the mock, but these reductions is statistically significant. And the panel F, G, E, F, G, H, I shows a H&E staining of the mouse models at different conditions. For the mock, we can see that the compactness is very high. However, for the case where T cell receptors only used, we see that there is a reduction in the compactness, and where there is only DiaBZl treatment, there is a further reduction in the compactness. And after that, if they use T cell receptors with DiaBZl treatment and the DiaBZls, they see that maximum reductions in the compactness. And when they only use this DiaBZls treatment, they see the reduction in the compactness, but compactness goes maximum in the case of panel G. And these things are quantified in panel J, where we can see that the reduction in the compactness is high, where T cell receptors which are treated with the DiaBZl and the DiaBZls are used in the mouse models that are affected with the injected with this MEL 526 cells. However, these things are compactness is minimum when only the T cell receptors is used. So only all this indicates that when the sting agonist DiaBZls, when it combined with this engineered T cell receptors, T cells, and these T cell receptors belongs to this 1G4 TCRT, then they will see, and these T cell receptors are also cultured in the DiaBZls based medium, then we will see the maximum reductions in the tumor growth and uh, tumor weight, or we can say that it will cause maximum regression of the tumors in the mouse models. So there, this combined effect will cause suppressions of the tumors, which is evident in these mouse model studies. So I hope you understood how they made a point that this combined immunotherapy will cause reductions or will impairs the tumor growth. So I hope you understand the basic mechanisms behind this, and if you want to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.